Imagine your body is the most complex, incredible machine ever built. It can run, jump, think, heal, and even create more machines just like it. Now, where would you find the instruction manual for such an amazing contraption? You don't have to look far, it's tucked away inside nearly every single cell in your body. This manual is called your DNA, and it contains all the information needed to build and operate you. Each chapter in this enormous book is a gene, and these genes are the specific instructions for everything from the color of your hair to how your heart beats. It's a blueprint, a recipe book, and a user guide all rolled into one, passed down through generations. Every living thing, from a towering redwood tree to a tiny ant, has its own version of this instruction manual. It's the universal language of life. This set of instructions, your genome, is written in a simple four-letter alphabet, A, T, C, G. These letters are arranged in a specific order to spell out the words, sentences, and chapters, our genes, that define us. This code is so detailed that if you were to print out the entire human genome, it would fill a stack of books as tall as a skyscraper. It's a mind-boggling amount of information, all packed into a space so small, you need a powerful microscope just to get a glimpse of it. This manual isn't just a static document, though. It's dynamic and interactive. Your body is constantly reading it, interpreting the instructions, and carrying them out. It's like a chef continuously consulting a recipe book to cook a magnificent, never-ending feast. The recipes, our genes, tell the cell what ingredients to use, proteins, and how to combine them to build everything from muscle fibers to the photoreceptors in your eyes that let you see the world. This process is happening right now, in trillions of cells all over your body, without you even having to think about it. It's the silent, tireless work that keeps you, well, you. Think of your body as a bustling city. The DNA in the nucleus of each cell is the central library, holding all the city's blueprints. When the city needs a new fire truck, a messenger called RNA goes to the library, makes a copy of the fire truck blueprint, the gene, and takes it to the factory, the ribosome. The factory then reads the copy and builds the fire truck, the protein, according to the exact specifications. This system is what makes life so diverse and complex. A muscle cell and a nerve cell both have the same complete set of blueprints in their libraries. However, the muscle cell focuses on building proteins like actin, myosin, which allow it to contract. The nerve cell, on the other hand, focuses on building proteins that create neurotransmitters, ion channels, allowing it to send electrical signals. It's not that they have different instructions, it's that they are reading different parts of the same master instruction manual. The control of which blueprints get read is one of the most fundamental secrets to how life works. It's truly amazing. So, if every cell has the same giant instruction book, how does a liver cell know to act like a liver cell and not a skin cell? This is where a fascinating process called gene regulation comes in. Gene regulation is, simply put, the process of turning genes on or off. A gene that is on is being actively read by the cell, its instructions are copied, and the protein it codes for is being made. A gene that is off is silent, it's still there in the DNA, but the cell is ignoring it so its protein isn't produced. Imagine your genome as a massive piano with tens of thousands of keys. Each key represents a gene. At any given moment, a cell is not playing all the keys at once. That would be chaos. Instead, it plays a specific melody, pressing only the keys, genes. It needs to perform its unique function. A heart cell plays a heart cell song, a brain cell plays a brain cell song, and so on. Gene regulation is the invisible musician deciding which keys to press and which to leave untouched. This allows for the incredible specialization we see in our bodies, where different cells perform vastly different jobs. This on and off switching isn't just a one-time thing, it's a dynamic process that responds to your environment, your age, and even what you eat. This constant adjustment is happening all the time, helping your body maintain a stable internal environment, a state we call homeostasis. It's a beautifully responsive system. To really get a handle on gene regulation, let's use some simple analogies. The most straightforward one is a light switch. Think of each gene in your DNA as a light bulb connected to a switch. When a cell needs to perform a certain task, it flips the switch for the corresponding gene to on. The light bulb turns on. The protein is made, illuminating the cell with its function. When the task is done, the cell flips the switch to off and the light goes out. Another great way to think about it is a giant factory, with thousands of different machines. The DNA is the central control room. Each gene is a blueprint for a specific product. You don't want every machine running all at once. That would be inefficient. It would waste energy. It would create a lot of products you don't need. Instead, you have factory managers, transcription factors, who go around and decide which machines to turn on, based on the current orders. If the factory gets an order for more red widgets, the manager activates the red widget machine. This manager is responding to signals from outside the factory, just like our cells respond to signals from our body and from the environment. We can even take the factory analogy a step further. Imagine the DNA is a tightly wound spool of thread. To read a gene, you have to unwind that specific section of the thread. The cell can use chemical tags to either keep the thread wound up tight, gene off, or loosen it to make it readable, gene on. These tags can be passed down when a cell divides, which is why a liver cell gives rise to more liver cells. They inherit the same pattern of on and off genes. This regulation isn't just about on or off, it can also be like a dimmer switch. Gene regulation allows the cell to fine-tune the level of protein production, turning the dimmer switch up or down as needed. It's an incredibly sophisticated system of management happening on a microscopic scale. Our DNA copying machinery is incredibly accurate, but it's not perfect. Imagine you had to copy a billion-letter book by hand, you'd probably make a few typos. The same thing can happen in our cells. A mutation is simply a change in the sequence of ATCGs in our DNA. It's like a typo or an editing mistake in the genetic instruction manual. These changes can happen when a cell makes a mistake while copying its DNA before dividing, or they can be caused by exposure to certain things in the environment, like ultraviolet radiation from the sun or some chemicals. Most of the time, our cells have amazing proofreading systems that find and fix these typos before they become permanent. But sometimes, a mistake slips through. This change can be as small as a single letter being swapped for another. This is called a point mutation. It's like changing one letter in a recipe. Instead of bake for 30 minutes, it might now say, bake for 80 minutes. Other times, the mistakes can be larger. A whole chunk of DNA might be deleted, duplicated, or inserted into the wrong place. These mutations happen randomly, and are happening in our bodies all the time. The vast majority of them occur in regular body cells, somatic cells, and only affect that individual. However, if a mutation occurs in the sperm or egg, germline cells, it can be passed down to the next generation. This is how genetic traits and inherited diseases are transmitted from parent to child. It's important to remember that the word mutation can sound scary, but it's a natural and fundamental part of biology. Mutations are the raw material for evolution. They create genetic variation, the differences that exist between individuals in a population. They are not just mistakes, they are possibilities. Not all mutations are created equal. Their effects can be sorted into three main categories. Bad, good, neutral. A bad or harmful mutation is a change in the DNA that causes the resulting protein to malfunction or not be produced at all. This can lead to a genetic disorder or disease. It's kind of like a typo that changes add 1 cup of flour to add 1 cup of salt. The resulting cake is going to be a disaster. Then there are good or beneficial mutations. For example, a long time ago, a mutation occurred in some humans that allowed them to continue producing the lactase enzyme into adulthood. This enzyme lets them digest milk. In populations that domesticated cattle, this was a huge advantage, providing a rich source of nutrition. This trait, called lactase persistence, spread rapidly. The vast majority of mutations, however, fall into the third category, neutral. A neutral mutation is a change that has no noticeable effect on the organism. The mutation might occur in a stretch of DNA that doesn't code for any protein, part of the non-coding regions of our genome. Or the change might be so small that it doesn't alter the function of the protein it codes for. The genetic code has some redundancy, so different three-letter words can sometimes code for the same amino acid. It's like changing the word big to large in a sentence, the meaning stays the same. So when you hear the word mutation, don't immediately think of something negative. Think of it as just a change. Most of the time, these changes are silent, slipping by unnoticed. Occasionally, they cause problems leading to diseases that scientists are working hard to understand and treat. And every once in a while, a mutation provides a surprising benefit that can change the course of a population's future. The interplay of these different outcomes is what drives the incredible diversity of life we see all around us, from the different patterns on a butterfly's wings to the different blood types in humans. Let's look at some real-life examples of how these genetic changes play out. One of the most visible examples of a harmless mutation is eye color. Originally, all humans had brown eyes. Between 6,000 and 10,000 years ago, A single person in Europe had a mutation in a gene called HERK2. HERK2 acts as a switch for another gene, OCA2. OCA2 controls production of brown pigment, melanin, in the iris. The mutation turned the switch to a dimmer setting, reducing melanin. With less melanin, the eyes appear blue. Every blue-eyed person today traces back to that one individual. On the other end are harmful mutations causing serious diseases. Consider cystic fibrosis. Cystic fibrosis is caused by mutations in the CFTR gene. CFTR makes a channel on cell surfaces. The channel lets chloride ions pass. This controls water movement in tissues. When mutated, the channel is missing or doesn't work. That causes thick, sticky mucus in the lungs and in the digestive tract, causing breathing and digestive problems. A single gene typo can devastate the whole body. Another example, sickle cell anemia. A single point mutation, one letter change, in the hemoglobin gene. This change makes hemoglobin stick together, deforming red blood cells into sickle shapes. Those cells can't carry oxygen well and clog small vessels, causing pain and organ damage and anemia. A tiny genetic change can massively affect health and life quality. Sickle cell can be both harmful and beneficial. 2 copies cause anemia. 1 copy gives protection against malaria common in parts of Africa and in other tropical regions. The malaria parasite struggles in sickle-shaped cells. In malaria-heavy environments, 1 copy is a survival advantage. Good or bad depends on the environment. Proper gene regulation is absolutely essential for keeping us healthy. It's the conductor of our cellular orchestra, ensuring that every instrument plays the right music at the right time. When this conductor falters, genes are turned on when they should be off or off when they should be on. Things can go very wrong. This misregulation is a root cause of many complex diseases, including cancer. Cancer is essentially a disease of uncontrolled cell growth, and this growth is often driven by faulty gene regulation. For example, our bodies have genes called proto-oncogenes, which help cells grow and divide in a controlled way. Think of them as the gas pedal in a car. We also have tumor suppressor genes which act as the brakes, telling cells to stop dividing or even to self-destruct if they become damaged. Cancer can occur when a mutation causes a proto-oncogene to get stuck in the on position, like a gas pedal stuck to the floor, or when a mutation disables a tumor suppressor gene, like the brakes failing. The cell loses its ability to follow normal rules and begins to divide uncontrollably, forming a tumor. Gene regulation is also critical during our development from a single cell into a complete human being. The precise timing of when genes are turned on and off guides the formation of our organs, limbs, brain. If a key developmental gene is activated too early, too late, or in the wrong place, it can lead to birth defects. The intricate dance of gene expression is what ensures your heart forms in your chest and not your foot, and that you have 5 fingers on each hand instead of 7. It's a process of astonishing precision, all orchestrated by these regulatory switches. Beyond development, ongoing gene regulation is key to our daily well-being. It helps our immune system respond to infections by turning on genes for antibodies and other defense molecules. It helps our bodies adapt to changes in diet, exercise, and even the time of day. Our internal body clock, or circadian rhythm, is controlled by a core set of clock genes that turn on and off in a roughly 24-hour cycle, influencing our sleep patterns, metabolism, and hormone levels. When this regulation is disrupted, it can contribute to sleep disorders, metabolic problems, and other health issues. Proper gene regulation is the silent guardian working around the clock to maintain order in our cells. The incredible knowledge we've gained about genes, regulation, and mutation is revolutionizing the field of medicine. By understanding the genetic basis of diseases, scientists and doctors are developing powerful new ways to diagnose, treat, and even prevent them. One of the most exciting frontiers is gene therapy. The idea is simple, though the execution is complex. If a person has a disease caused by a faulty gene, why not try to fix that gene? Why not replace that gene? It's kind of like finding the typo in the instruction manual and correcting it with genetic whiteout. Scientists are already having success with this approach. For certain inherited diseases, some forms of blindness, severe combined immunodeficiency, researchers have used harmless viruses as delivery vehicles to carry a correct copy of the faulty gene into the patient's cells. Once inside, the correct gene can start producing the functional protein, potentially curing the disease, dramatically improving the patient's symptoms. This is a personalized, precise form of medicine that targets the root cause of the problem, not just the symptoms. It's a complete game changer. Understanding gene regulation is also opening doors to new kinds of drugs. Instead of just treating the symptoms of a disease, what if we could develop drugs that specifically target the faulty gene switches? For example, if a cancer is being driven by a gene that's stuck in the on position, perhaps we could design a molecule that flips that switch back to off. Researchers are actively developing these kinds of epigenetic drugs that can add chemical tags, that can remove chemical tags, that control gene accessibility, essentially reminding the cancer cells how to behave properly. This offers a more targeted and potentially less toxic alternative to traditional chemotherapy. Furthermore, our growing knowledge of the human genome is ushering in an era of personalized medicine. By sequencing an individual's genome, doctors can identify genetic variations that might make them more susceptible to certain diseases, heart disease, diabetes. With this information, they can recommend personalized lifestyle changes, preventative screenings. It can also help predict how a person will respond to certain medications, allowing doctors to choose the most effective drug and dosage from the start, avoiding a painful and costly trial and error process. Understanding our own genetic code can empower us and our doctors to make smarter, more proactive decisions about our health, shaping a future where medicine is tailored specifically to you. Consider the possibilities.